A very good morning to all of you. I welcome you all to my channel Studika Enzyme, and today we are here continuing with the part 2 of Principles of Inheritance and Variation, Class 12 Biology, chapter number 5. So in the previous part 1 of this chapter, we had discussed about the monohybrid crosses. I think some of you had little doubts about how to fill up the Punnett square. So we will continue with this Punnett square filling and continue further in this slide. So let's proceed. So as you can see, this is a Punnett square with one square and four squares inside that, and in this you will be putting up the genotypes on both the sides, as you can see in this box number 2, and then you will split, like law of segregation says, splitting of the alleles before they go into the gametes. So these genes, these alleles, they are separated from each other, and then you put them here, as you can see, and then as we fill up the matrix, we keep on crossing and then filling them, right? So this is how we fill the Punnett square, and then we get the genotypes. Now here we have a dihybrid cross where two traits are involved. As we saw in the monohybrid cross, only one gene was talked about. For example, tall and dwarf genes, yellow seed color, green seed color, round seed, wrinkled seed. But when we talk of two genes together, as you can see here, round seed and yellow seed. The other allele could be wrinkled and the green seed. When we see their inheritance, their cross, then we call it a dihybrid cross. And for all the F1 generation here will be all round and yellow. I will show you this cross. And the F2 ratio always have to be 9:3:3:1. And we will be able to see this law of independent assortment also here. As you can see on the screen, this is one allele, round and yellow seed. This is the other one, two genes, green and wrinkled. So when these are crossed, so we write them like this. So I would be writing it somewhat like round, I'll use a different color, round, and yellow. Since round and yellow are dominant, so we will use the capital letters for these, and wrinkled we will use small r, and green we will use small y. So first of all, law of segregation says that these are to be separated. So they will be separated like ry. This will be separated like ry, as you can see here. So these are the gametes which can on fertilization be fusing to form the to form the zygotes. So when these two fuse, to form a zygote, which is capital R, capital Y, small y, which is heterozygous, round and yellow. Heterozygous because it has two different alleles, capital R and small r, and the phenotype will be round and yellow only for all the F1 generation that you can see here. Now on selfing of F1 generation, as we did in the monohybrid cross also, on selfing of this F1 generation, we get four new combination of genes: capital RY, capital R, capital Y, capital R, small r, capital Y, small y. So when these are crossed among themselves, when these will be crossed, so now you get a Punnett square somewhat like this. So you will have, and when you will check it, you will be able to see four different combinations: round and yellow 9, round and green 3, wrinkled and yellow 3, wrinkled and green 1. So this ratio always have to be the same ratio. So we call it dihybrid ka F2 ratio phenotypic will be 9:3:3:1. Okay, but one thing I would want you all to notice here, initially what was the combination of genes that we used? Yes, the initial combination of genes that we studied was round yellow on one side, wrinkled and green on the other side. I mean, one parent was round and yellow and the other parent was wrinkled and green. But when this uh, hybrid or this cross began and we reached up to F2 generation, so you will be able to see that we have two new combinations, round and green, wrinkled and yellow, which were not there in the previous. So these are new non-parental combinations. So this shows one thing, that round gene, which was initially with the yellow gene, now is found with green also. This wrinkled, which was initially with the green, is now with yellow also. So this means that genes will also keep on changing their position. Genes will keep on switching the partner genes. What does that mean? Suppose एक प्लांट था पी का, ठीक है, जिसमें राउंड और रिंकल्ड जीन्स दोनों साथ साथ थी. This was round and this was round and chalo yellow. Gene was together. And in the other combination, other parent, you saw wrinkled and yellow. But now you have four combinations, right? So one combination is like parental only RY. The other combination is the parental only small r, small y. But now you will see this new combination, capital R and small r and capital Y. So these two new combinations will show that the genes which were initially together can also change their position. So this is law of segregation. Law of segregation tells us that two genes which were initially together on one chromosome, if they are little apart from each other, so they can form new combinations in the next generation. Okay, so now we have understood both monohybrid and dihybrid cross. Let us recap all three combinations, all three laws of inheritance. So first law of inheritance is law of dominance. So law of dominance states that one allele will be dominant over the other. So one allele will express itself over the other one. Why? For example, a tall gene and a dwarf. So tall is dominant over the dwarf. As you can see on the screen, brown eye color is dominant over the blue eye color. So if an individual has one allele for brown, another allele for blue, so he or she will have brown eyes only, because brown is dominant over the blue eye color. Now how does that matter? How is this possible? This is due to the functional gene formation. So a dominant allele will form completely functional and complete protein which is responsible for one trait. But in case of recessive alleles, the protein which is formed is either incomplete or less efficient, because of which the protein which is formed for that particular trait is not complete. Therefore, they have a modified appearance or a different appearance from the dominant one. Okay. Moving on to the second law of inheritance, this is law of segregation. Law of segregation states that two alleles which were together in an individual, they separate during the formation of gametes. And these gametes so law of segregation that we have seen, these gametes, they will have the two different alleles or the two same alleles of a gene. This is law of segregation. Third law is the law of independent assortment. Now, independent assortment, we have just now seen that the two genes which are there together in the parental combination, they may change. Initially, that you can see here, capital A, capital B were together, but in the offspring, but in the F2 generation, you can see capital A with small b, capital A with small b, small a with capital B. So these are the new combinations that are formed, right? So this is law of independent assortment. Like Genes assort themselves, separate themselves independent of the neighboring allele or the neighboring gene. So these are all three laws of inheritance. Um, Children, I would also want to tell you one thing. So these are all Mendelian laws of inheritance, but there are different inheritance where Mendelian laws are not followed. So we call them non-Mendelian inheritance, and they are very important from board point of view. So these are usually asked in grade 12, because you have read your Mendelian mono and dihybrid in your grade 10 also in chapter heredity and evolution. Here we will begin with non-Mendelian inheritance. First one is incomplete dominance. Now what do we mean by that? Incomplete dominance, we will study uh, ahead. So incomplete dominance states that the dominant allele is not completely able to mask itself over the recessive one. So as a result of this, the heterozygous state that we get will have intermediate structure. So you have seen in case of a pea plant that the flower can be violet or it can be white, but it will never be light violet, it will never be some other combination of that. But in case of snapdragon plant, in case of Mirabilis jalapa, you will be able to see when red color, when one parent has red colored homozygous condition and the other one is homozygous for white, the F1 generation, which is capital R small r, will show, according to Mendelian inheritance, it should show red color. But in case of snapdragon, this color has to be pink, which is a combination of white and red. So the red dominant allele is not able to mask itself completely. So we see a new phenotype in case of a incomplete dominance. So the phenotypic ratio will become 1:2:1. So one red, two pink, and one white. So this question can also be asked in a board examination. Which type of dominance, which type of inheritance is it in which you see the phenotypic ratio to be 1:2:1? You have to remember it very clearly that the genotypic ratio in case of Mendelian inheritance is 1:2:1, but phenotypic ratio is always 3:1. But in case of incomplete dominance, it is 1:2:1. So that's incomplete dominance. Moving on ahead will be codominance. As the name suggests, codominance. अब क्या है, दोनों alleles dominant की तरह behave करेंगे. So neither is able to mask itself over the other one. अगर हम human blood group की बात करते हैं, so human blood group will have three alleles, A, B and O. IA, IO and IB. So these three alleles, out of these three, A and B alleles are codominant. Now what do we mean by that? Codominant का मतलब हो गया दोनों ही equally dominant हैं. So it means अगर एक individual है जिसके पास IA और IB allele है, ठीक है, one allele from A blood group, one allele from B blood group. So he will not have either A or B. He will have a new combination of blood group which is AB. Okay, so both are able to express themselves equally. But they are, both of them are equally dominant over the O allele. If I talk like this, IA, suppose an individual has IA and IO gene. So A is dominant over O. So he will have blood group A, as you can see here. अगर ये genotype है AA, so he will have blood group A. AO, he will have blood group A. So if it has OO, so he will have blood group O. AB, so blood group will be AB. Instead of being A or B, it is a new combination altogether. So this type of inheritance is called codominance. Next, you have multiple allelism. Now, multiple allelism, as the name suggests, as you can see on the screen, the different skin colors of individuals existing on the globe. So, human skin color is also because of a number of alleles. So these alleles are responsible when we have more than one allele for one particular gene, then we call it multiple allelism. As you see in case of blood group also, we have three different alleles, IA, IB and IO. So these are three different alleles. So this condition is called multiple allelism. Then you have pleiotropy, which is very, very important. Pleiotropy means one gene leading to more than one impact. A single gene that you can see leading to three different traits, three different characteristics, three different impact or phenotype. So this is called pleiotropy. For example, sickle cell anemia also, and what you can see on the screen is starch synthesis in case of a pea seed. So the gene responsible is blue, the gene is B. When it is in the homozygous condition, okay, it produces large starch grains and large seed. So grains are also large and seed size is also large, right? But in case of homozygous small b, the size is small and the grain shape is wrinkled now. In case of heterozygous condition, so it is intermediate, right? It is intermediate. So we can also put it in the example of incomplete dominance also. So intermediate and intermediate size of seed, right? So this is an example of pleiotropy as well as incomplete dominance. That the same thing that we have discussed just now. Okay, then you have polygenic inheritance. Now what do we mean by polygenic? The name is clearly explaining itself. Polygenic means caused by many genes. When many genes are responsible for one particular trait, for example, human height, it is dependent on so many genes, right? Human skin color, that's also dependent on so many genes. IQ of an individual. So one gene is not responsible. Rather, a group of genes responsible for one particular trait is known as polygenic inheritance. And the examples are height of an individual, IQ, skin color. That's it.